So you may have noticed that I look a little rough, and that's because I have exams. I have exams in biology and calculus, and I know nothing. So I am going to read my notes as a podcast to you. Um, if anybody watches this, this is like crazy. Like, what are you doing with your time? Studying in a better way than just passively listening to somebody talk to you. But if you need explanation for anything, I have it. So we'll go unit by unit, and if I say anything wrong, please correct me in the comments. I won't be offended. Yeah. So one of the key, and probably the key building block of life is water, and water is has solubility, which is one of the key components of water. It's basically how easily a solute dissolves to a solvent. So like dissociation, saturation, and temperature are all key words involving that. If you don't know what they mean, look them up. Um, Water flushes waste systems, medications are soluble, and partial positives in water will attract chlorine, for instance, and partial negatives will attract sodium, for instance, and then that's how things dissolve into water when water isn't necessarily causing ionic bonds, but it's causing dipole charges. There's also cohesion and adhesion, which is cohesion is sticking to itself and adhesion is sticking to other materials, which water has both. Water molecules are slightly attracted to other materials and itself due to partial dipole charges, and it allows for smooth transportation of blood cells. So if you think about it like the blood cells, yeah, you get it. And then water has a high specific heat capacity. Specific heat is the amount of energy required to raise the temperature of 1 degree Celsius. Um, Vaporization is the amount of energy required to change liquid into a gas. Thermoregulation is the high heat capacity of water, which means temperature changes don't affect water as much. And then habitable bodies of water provide stable temperature. So strong hydrogen bonds means it requires more energy to change the temperature. So yeah. Now we move on to organic chemistry. Um, Organic molecules are based around carbon and hydrogen. Macromolecules are considered organic molecules. So examples of macromolecules are carbohydrates, nucleic acids, lipids, and proteins. But all these have one thing in common, and that is carbon. Carbon is frequently referred to as the building block of life, but why is that the case? Because it has 4 electrons, so it can gain or lose 4 electrons, meaning it can form 4 bonds, and these bonds can be covalent. So there are various types of covalent bonds: polar, nonpolar, single, double bonded, and carbon can create all of these. It also has small atomic radius, so it has lower energy bonds, and bonds with lower energy will remain in stable states. So just for some examples of what carbon can create, it can create hydrocarbons, which are just a bunch of carbons in a chain, and then hydrogens are attached to every single carbon, which will not dissolve. It can form chains, it can form rings with alternating double bonds. It can form branch chains with like carbon sticking out of nowhere from the regular zigzag. And then just so you know, like hydroxyls attached to a carbon ring will dissolve, but hydrocarbons will not. And you may be asking, what's a hydroxyl? A hydroxyl is a type of functional group, which you never split up a functional group. So basically, the way it works is there's something called an R group, which is everything else, and then there's these functional groups that they're attached to. So from the R group, you have an oxygen and a hydrogen. That's called a hydroxyl. That creates alcohols, which are polar soluble and written as a chemical shorthand of OH. Then you can have a carbon double bonded to an oxygen and then also bonded to a regular oxygen and a hydrogen attached to that oxygen, which are carboxyls, which form carboxylic acids or organic acids, which end in ic acid or ate. They're weak acids. They're polar but also ionic because they're acids. They're water soluble and they dissociate. The shorthand is COOH, COOH. Then there are R groups attached to a carbon attached to a double bonded oxygen up top, and hydrogen to the right. That is a carbonyl because it creates an aldehyde, which is weakly polar and somewhat soluble. But it can also create um, a ketone, which is a carbon double bonded to an oxygen, double bond, regular bonded, and the carbon double bonded to an oxygen, and the carbon is attached to another R group. So it's like in the middle of something. That's a ketone, which smells strongly, but it's weakly polar and somewhat soluble. So the shorthands for that is CHO or CO. Then there's an R group attached to an N attached to any amount of H's or even no H's sometimes. That's an amino. Basically, if you see a nitrogen, it's an amino. That creates amines, which are polar or ionic, basic because they have lone pairs on the nitrogen that can attract a hydrogen. So they're proton acceptors. Remember, that's the definition of an acid if you took chemistry last year. The shorthand is anything with an N. It could be NH2, NH, NH3 plus, N. Yeah. Then there's sulfhydryls, which are sulfur and a hydrogen. So basically, if you see a sulfur, it's a sulfhydryl. That creates thiols, which are nonpolar and they have a rotten egg smell. If you're familiar with the skunks, you should be. Skunks have sulfhydryl in their spray. The shorthand is SH. And then there are phosphates, which is a phosphorus with a ring of oxygens around, and one of the oxygens is attached to the R group, and another oxygen on the top is double bonded. That creates phosphates, which are ionic. The shorthand is PO42-, or P with a circle around it. Then there's molecular formulas, which have the elements, no structure shown, and are in the order of CHNOPS. Then there's structural formulas, which are like the full drawing of everything. And then there's skeletal formulas, which you can shorthand things. Change of direction is just like a hat. It's like a zigzag. And you don't write every carbon, you don't write every hydrogen, you just assume the hydrogens are attached to the carbons when you don't write anything beside them. And yeah. So then you may be asking, what's a polymer? That's the other part of this unit. Yep, I forgot about it too. Polymers are like not formed by lipids, but they're a long repetitive chain of monomers linked by covalent bonds. They can create monosaccharides, amino acids, or nucleotides, but not lipids. Remember that. A monomer basically stands for one unit. Mer means unit, mono means one. That's why polymer means many units, because mer means unit and poly means many. But monomers are joined by condensation synthesis, which I'll throw up on the screen, but it's like one type is called dehydration synthesis. And that's the one that we focus on in grade 12 bio. It's when they take the hydroxyl and the hydrogen and they join them together into water, joining together two monomers into a dimer. And then there's hydrolysis, which is when water is inserted back in basically the reverse of dehydration synthesis. And then that creates a polysaccharide, a polymer, and a monomer, or a polymer, a polymer and a monomer. Basically just biological polymers get broken apart by hydrolysis. And this is all done by carbohydrates, proteins, nucleic acids, but lipids do not form polymers. Then there's carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen, just like most other organic molecules, typically in a 1 to 2 to 1 ratio. They're suitable for energy storage due to high volume of carbon to carbon and carbon to hydrogen bonds. Electrons in bonds are transferred to oxygen, which energy is given, producing ATP, which we will get to later. Cellulose and chitin are examples. There are lots of hydroxyls and aldehydes and ketones. So some important properties are the many hydroxyls, because due to the polar nature of hydroxyls, the solubility increases, and solubility is important for carbohydrates as they go through the blood, they go through all these different things, all these different mediums. Then there's carbon and carbon and carbon and hydrogen bonds, because carbon and carbon bonds form the chain and of the backbone of the carbohydrate, while the carbon and hydrogen bonds hold energy that gets released when broken. For grade 12 bio, we focused on triose monosaccharides and pentose monosaccharides and hexose monosaccharides. Those are the only options for us. I don't know about your school. Monosaccharides are small, water soluble because they can dissolve better because they have a polar outside, so they're easier to transport to the blood. They're rich in chemical energy, usually between 3 to 6 carbons are present, dominated with hydroxyl groups, like there's so many hydroxyls. They enter through the food chain, and autotrophs store energy usually in the form of monosaccharides. So autotrophs are like things that make their own energy. Usually end in ose or glyceraldehyde. In this case, it's just glyceraldehyde. And then the rings form in the water. So there are isomers, they're structural isomers, which are different arrangements of the same unit. And then there's stereoisomers, which is the same arrangement but different orientations of the same unit. I'll throw up examples, but the most important one is alpha glucose or beta glucose. Alpha glucose contains a hydroxyl on the bottom, and beta glucose contains a hydroxyl on the top. That is a structural isomer. So disaccharides have 2 rings, not 1, but 2. You list them as in disaccharides of 2 hexose monosaccharides, for instance. And you're usually told if it joins from 1 to 4 or 1 to 2, or like what carbons are joining. Hydroxyl groups join and form linking oxygen and H2O, which is a glycosidic bond, which is formed by, you guessed it, dehydration synthesis. It stores 2 times the energy and because it's double the size, but has less osmotic potential because of less magnitude of osmotic gradient because of its size. It's not common in humans, and you don't draw beta glucose side by side. Draw it offset from each other like this. Polysaccharides, they're hundreds or thousands of monosaccharides long. They're for long-term storage because they're insoluble because they're so long. Plants and algae will string together alpha glucose in spiral and helical chains of amylose or amylopectin. And yeah, let's get into the 5 main polymers. So there are 3 alpha glucose types, and there are 2 beta glucose types. Plants and algae string together in a form called amylose. String glucose together in a form called amylose. And when a branch is formed, that is called amylopectin. Then amylose and amylopectin can form plant starch together, but that's not really important. What's more important is if it has so many branches that you can't count them all, or you can count them all, but there's just a lot of branches, that will be glycogen. Animals will make or store glycogen, but plants will not. Plants will make amylose, amylopectin, or plant starch. Then there's cellulose or fiber, which is beta glucose 1,4. It supports plants and cell walls, and it's undigestible by humans. So that's why like fiber bulks up your fecal matter, because you can't digest it. So it's just bulking everything up. And then there's chitin, which is glucosamine. It's a modified carbonyl with an amino, and it's polar, which allows the number of hydrogen bonds to go up. Chitin kind of like strengthens exoskeletons and stuff like that. So then we have lipids. What are lipids? They're made of carbon, hydrogen, oxygen as well. There are way more carbon and hydrogen bonds than anything else. Many lipids also have ester bonds, which is carbon attached to oxygen attached to carbon, double bonded to an oxygen. They are very nonpolar, so they're insoluble in water, soluble in oil. Their functions are they're waterproof because they're hydrophobic, so useful whenever you need waterproofing for like cell membranes and oils, phospholipid bilayer. They protect vital organs, and they're an insulator for temperature and electricity. And they store energy, 2.5 times the energy in lipids than carbs. Then there are many types of lipids, but the main ones are triglycerides, which are fats and oils mainly for energy storage and waterproofing. They range from 4 to 8 carbons in each fatty acid. There are phospholipids, which are waterproof in cell membrane They have a region that are polar and a region that are non-polar, so they're amphiphilic. These ones have like two fatty acids, a glycerol, a phosphate, and then something polar. That's what makes membranes. Then there's waxes. Then there's waxes, which are an ester bond formed by a long alcohol bonded to a fatty acid. And a fatty acid, which we've been talking about, but you don't know what that is because I haven't defined that yet. Or ideally you know what that is because it's exam season. But they're named fatty because they're long, nonpolar, and acid because their functional group is at the end. So there are saturated fatty acids, which are like a bunch of carbons in a chain and then hydrogens attached, no double bonds. All the carbons have hydrogens on them. And there's unsaturated fatty acids, which have a bunch of hydrogens, a bunch of carbons, but some of the carbons are double bonded because there's not enough hydrogens to saturate them. These are like the building blocks of lipids. Like that's the main form they're found in. And then there's steroids, which are hormones for cell-to-cell communication. Cholesterol is one, and it's the precursor for a lot of them. They contain no ester bonds, no phosphates, and many have double bonds somewhere. They're found in like rings with each other. So just for a recap, saturated and unsaturated fatty acids, when you're naming them, can be used for triglycerides, phospholipids, and fatty acids. And then monosaturated, diunsaturated, polyunsaturated is what you say when you're talking about fatty acids. And then you call it a wax if it's a wax. That's it. And then proteins and nucleic acids. Proteins, there are structural proteins that create like the cytoskeleton, and then there's functional proteins which are for mobility, transport, immunity, enzymes, hormones, acid-base regulation, all of that stuff. Not important what they actually do, just know they do things. Proteins are polymers of amino acids, monomers, that are three-dimensional and made up of elements C, H, and OS. There are only two monomers that contain S, though, just so you know. There are 20 types of amino acids, but they all kind of follow the same structure. There is a amino on the left. There is an R group attached on the top. There's a carbon in the middle. There's a hydrogen at the bottom, and there's a carboxyl on the right. And then the R groups that I'm talking about, there are four main types of these R groups. There are non-polar R groups, which contain lots of CHs. There are polar R groups, which contain non-charged polar bonds. Aminos are basic with H3, just so you know. So uncharged will be polar. Tyrosine's in this category because of the OH on the end, but don't worry too much about that. There's acidic electrically charged, which contain charged hydroxyls, and there's a basic electrically charged, contain charged aminos. So remember, that's the H3. And then there's polymerization, which creates a peptide bond. That's when basically the protein goes through condensation synthesis. And then there's primary structures of proteins, which is just a polypeptide, so a string of amino acids, and then R groups sticking out at the edges, but the R groups don't attach to anything. There's secondary structure, which is the hydrogen bonding of the peptide backbone to itself, causes amino acids to fold and repeat in the pattern. So there's alpha helix structures, and beta pleated sheet structures, which are like zigzag sheets. Then there's tertiary structure, which is 3D folding due to R group interactions. Some proteins will just stop here. This is where enzymes can start to be. This is where, yeah. And then there's quaternary structure. So not all proteins will get here. Some just stay at tertiary. But that's 2+ polypeptides attaching through hydrogen bonding. So multi-enzyme complexes are grouped quaternaries, and they're basically just enzymes that are more effective. Now you may be wondering what's linking those together. That's tertiary structure. That's hydrogen bonds, disulfide bridges, ionic bonds, van der Waals attractions, and hydrophobic exclusion. Please Google them. I have absolutely no notes written down about them. And then I mentioned the word enzyme a lot, but what is an enzyme? Most enzymes are proteins, All enzymes are examples of organic molecules known as proteins that catalyze biochemical reactions within a cell. A substrate is specific to activate each enzyme, and basically, an enzyme is to a substrate what a lock is to a key. And that's basically all you need to know, except that there are ideal conditions for each enzyme that vary. So there's pH and there's temperature. If they're not in their ideal conditions, they are less effective, or they can denature, which basically means they're expired, they're dead, etc. If you get it. Now you may be asking, what's nucleic acid? Because I was talking about that a bit earlier. Um, Nucleic acids are molecules that contain CHNOP. Definitely it's a nucleic acid if it contains nitrogen or phosphorus. It has no carboxyls, and it codes information for one structure of protein. So basically, nucleic acids are made up of nucleotides. Nucleotides contain a phosphate group, a pentose monosaccharide, hydrogen if it's DNA, or a hydroxyl if it's creating RNA at the 2' location, and a nitrogenous base, which there are 5 options for. There's purines that create these nitrogenous bases, which are adenine and guanine, and there's pyrimidines, which have a single ring structure. So there's purines will have a double ring structure, and then pyrimidines will have a single ring structure, and these single ring structure ones are cytosine or uracil in RNA, or cytosine and thymine in DNA. So what's DNA? DNA is deoxyribonucleic acid. It contains AGCT as its bases. It is strung together phosphodiester bonds, then hydrogen bonding a second string together. The bases will hydrogen bond together to form a double helix. But to make sure that this happens, they must be anti-parallel. Like the strands must be anti-parallel, which basically means one must be upside down. And then these go through complementary base pairing, which is a specific purine and a specific pyrimidine bonded together. So that's A and T and G and C. And then RNA is ribonucleic acid, which is AGCU. It is strung together phosphodiester bonds as long as it needs to be, but it's a single helix. Just so you know, purine monomer nucleotides as monomers have functions. So RNA A, so RNA adenine is a part of ATP. So adenosine triphosphate, I believe. And yeah, that is biochemistry. So now we move on to molecular genetics, I believe. And basically, who discovered DNA? I don't know. But I do know. Friedrich Meister. Meister? I don't actually know how to pronounce that. He discovered DNA and found different levels of nitrogen and phosphorus and called it the nuclein, thinking it was a new substance. Then there's Phoebus Levene in the early 1900s who discovered bases AGTC, deoxyribose, and phosphate. He called them nucleotides, but believed in tetranucleotides, which is wrong. Then there's Frederick Griffith, hinted at the function of DNA in 1928. Bacterial transformation, something from converting live form. It was confusing. Avery McLeod McCarthy identified nucleus in DNA was responsible for transformation. And then Hershey and Chase. And Chase, guess what? Was a woman. There was a woman on the team. Yes. Information confirmation saying that the DNA equals instructions. So DNA reprograms cells. And then Erwin Chagraf. He's not important. I didn't highlight anything. Then we get to Wilkins and Franklin. And then they made the Watson and Crick thing and the double helix structure. And then Watson and Crick stole the information from the woman that I forget the name of. It'll come to me. Rosalind Franklin. They stole her idea of the 3 different ways of replication. They didn't steal this. They stole the other stuff. But they said there were 3 different ways of replication: conservative replication, intact molecules; dispersive replication, chopped by enzymes; and semi-conservative replication, where one stands intact. And then the Mielsen and Stahl experiment of 1958 supported the semi-conservative molecule model. but it said not conservative replication of the first trial. And then the second round provided DNA replication is semi-conservative. This is the one where they shot the light through the test tubes, and the light for the 14 nitrogen and the heavy 15 nitrogen, and then everything ended up being either light nitrogen or in the middle. And yeah, you get the idea. But to understand this, we have to kind of understand DNA replication, and the enzymes that start it. So in DNA replication, there's an enzyme helicase, which unwinds and separates the DNA double helix, but not the phosphodiester bonds. Then there's DNA primase, which builds a sequence of RNA bases so DNA replication can start. This is all done at the origin of replication, called the replicon, with A and T pairs, with like a bundle of A and T pairs because they're easier to break because there's only 2 hydrogen bonds instead of 3 hydrogen bonds like there is for G and C. Then the SSBs, or the single strand binding proteins, keep the bubble at the replicon open. Then the DNA polymerase 3 synthesizes DNA on the leading strand, the continuous strand, and the lagging strand, which is the discontinuous because it has the Okazaki fragments, because not, yeah. And then DNA polymerase 1 creates hydrolysis on phosphodiester bonds to remove primers and add DNA. And then DNA ligase attaches fragments using ATP, and that's it. That's all of replication. So DNA is found in the nucleus, and RNA is found in both the nucleus and the cytoplasm, and then protein is found in cytoplasm only. How does that work? How does that make sense? Basically, RNA is a less important and less precious, not less important, but less precious form of DNA. So it goes into the cytoplasm to go do things. RNA polymerase 2 adds in the 5' to 3' direction from the promoter region, which is 25 bases upstream, along with help from transcription factors. And then the AAU-AAA is 10 to 35 nucleotides down. Then from the AAU-AAA, which is the stop, 10 to 35 nucleotides down, RNA polymerase 2 will fall off the strand. And then there is a 5' cap and a 3' poly A tail added, and 3' poly A tail keeps RNA around in the cytoplasm longer, depending on how long it is. And this RNA polymerase 2 is hydrogen bonded to the template strands. The template strands is the bottom one, and the coding strand is the top one, and then the new RNA strand is attached to the template strand through hydrogen bonds, is basically what that means. And then RNA splicing will cut out the introns because they don't code for proteins, and SNRPs will connect exons and create mature mRNA. So one codon is 3 bases, and this one codon codes for 1 amino acid. And also recall the central dogma of biology is DNA to RNA to proteins through transcription and translation. So mutations are accounted for through the redundancy of multiple codes creating the same amino acid. There are 2 regions of charged tRNAs, which if you remember the charging reaction, it's like very long-winded, but I have it here and I'll explain it anyways. So basically the amino acid attaches to the tRNA and the amino acyl in the tRNA. That's one thing. And the amino acid reacts with ATP to the carboxyl end of the amino, attaching to the AMP. The amino acid and AMP binds to the enzyme, then tRNA binds to enzyme, then transfers amino acid to tRNA, producing charged tRNA and AMP. And the amino acid is attached to the 3' acceptor stem. Basically, it makes no sense, but we'll get it. I'll put up an image. I just took my notes. There are 2 regions of charged tRNA with the acceptor end and the anticodon loop. The anticodon loop is complementary with bases to the codon, so the codon will hydrogen bond to the anticodon loop, and then proteins bond to the acceptor end. Ribosomes, the site of translation, are made of a large subunit and a small subunit that sandwiches a mature mRNA. They have A sites, which bind to tRNA with new amino acids, like the next amino acids. P sites, which has the polypeptide chain, and an E site, which binds to the tRNA that has a previous amino acid. So it's the exit site, like E stands for exit. So the steps for going through this means one, the rRNA in the ribosome binds to the mRNA and you have a start codon, and the small subunit then scans for AUG, which is methionine, which is the start codon. Then the initiator tRNA binds to AUG in the P site. Then the ribosome assembles. So this small subunit and the large subunit, they assemble, and hydrolysis happens between the amino acids, and two amino acids connect. Then the ribosomes move and the codon to the right, move one codon to the right. So A to the P site and then P to the E site. The amino acid will go on top of the single amino acid on the in the A site, not like the single one going on top of the chain of 100. No, the chain of the 100 will go on top of the single. And then this repeats until we reach the release factor, which binds to the A site of the ribosome. Polypeptide chain releases, then the ribosome dissociates from the tRNA. If the protein is for inside the cell, then this will only happen, like nothing else will happen. But if it's exiting the cell, the ribosome will dock on the endoplasmic reticulum for folding. The disulfide bond will form. For folding, disulfide bond formation, quality control, and glycosylation. And then proteins are sent through a vesicle to the Golgi apparatus, where the Golgi will modify and package proteins for leaving the cell. And guess what? That's molecular genetics. So what is cellular respiration? Cellular respiration is sugar, or glucose in this case, so C6H12O6, plus 6O2, or oxygen, turns into 6CO2 plus 6H2O, but in reality it's 12H2O because H2O is consumed, but that's the net H2O, become an exergonic reaction. So it's like negative 2870 kilojoules per mole. What's the point? It's to get ATP out of molecules such as glucose. So I mentioned the word exergonic and endergonic. Exergonic is like you start up here and then you go up and then you go down and you end up losing energy. So there's a negative delta G. So cellular respiration, or in this case, cellular respiration is exergonic. And then there's endergonic, which is you gain energy in the process. So that's ADP plus phosphorus. You'd start below and then you have a big activation energy, and then you plateau high up, that producing ATP, which is an endergonic reaction. So there's a positive delta G. So, and then we remember anabolic and catabolic. Anabolics building up of small parts, catabolics breaking down small parts. So glycolysis is where we start. We start with glycolysis. Please say you've seen that. In the cytoplasm. It means sugar breaking. It's 10 sets of reactions with enzymes catalyzing reactions. There's a small profit, so there's 2 molecules of ATP versus for total cell respiration, there's 36 molecules of ATP. And there's a lot of redox reactions, so that's oxidation and reduction. So losing electrons is oxidized, gaining electrons is reduced. The donor loses energy, receiving gains energy, so it's high energy, so it's further from the nucleus. High energy levels, right? Further from the nucleus. And then reduction reactions, so NAD plus to NADH plus H plus. So 2 electrons carried from 2H plus NAD, breaks 1 double bond, neutralizes charge of N plus, then dissociates. And this is done with like coenzymes and energy carrier molecules and electron carrier molecules. We'll get into that. And then there's substrate level phosphorylation, which is enzyme and PEP substrate, plus ADP, which is removal, which is exergonic, and transfer, endergonic, of phosphate from PEP into ADP. And without the phosphorus, it turns into pyruvate, not PEP. So, how does it start? We start off with 6-carbon glucose molecule, and then ATP turns into ADP. We turn into a glucose with a phosphate attached. Then more ATP turns into ADP. So now we're at 2 ATP used up, and we have fructose 1,6 bisphosphate, which is a 2-phosphate 6-carbon molecule. From there, cleavage occurs. So we have 2 3-carbon molecules with 1 phosphate attached. And then we use NAD plus to turn into NAD plus H plus, and we add a random phosphate. It just comes in for our purposes. It'll just appear. And then we appear with 2 phosphates and 3 carbons for bisphosphate glycerate. And then ADP turns to ATP, and we end up with 3 PG and 1 ADP produced, or 2 ATP reduced, because we have this happening twice simultaneously, and a phosphate attached. Then it turns into 2 PG, which is a 1 phosphate, 3-carbon molecule, then BP, then PEP, sorry. So same thing, by substrate level phosphorylation. This is happening, and then more substrate level phosphorylation, ATP turns into ADP turns into ATP, and we get pyruvate as a result. So the 2 NADH plus H pluses I mentioned go into the center and then of the cytoplasm, right by the outer membrane and intermembrane space of mitochondrion, and they turn into from 2 NADH plus H plus into 2 NAD plus. And then the 4 hydrogen electrons, the 4 hydrogen protons that were taken off, actually go to 2 FAD inside the mitochondrion and turn that into 2 FADH2. That is all glycolysis. That's all it is. Then we move on to pyruvate oxidation. I didn't write anything in my notebook. We start with a 1-carbon molecule, CO2. No, sorry. We start with CO2 being released and 1 NAD plus turning to NAD plus H plus H plus, and acetyl-CoA, which is a 2-carbon molecule with coenzyme A attached. So we just added 2 things and then released 2 things. So we released CO2 and NADH, and we added NAD plus and coenzyme A. And that is pyruvate oxidation. That's the only part of that reaction. But then we go into the Krebs cycle, the lovely, lovely Krebs cycle. I think there's a video of this guy doing a Krebs cycle rap. Like, it's the Krebs. It's the Krebs. So yeah, go watch his video. He's really good. But basically, we have citric acid. We have oxaloacetate, a 4-carbon molecule, added to the acetyl-CoA, and that turns it into citrate. Citrate will release CO2, and NAD plus will turn into NADH, and then we will end up with alpha ketoglutarate, which is a 5-carbon molecule because we released 1 carbon from the CO2, and then we release another carbon molecule through CO2. Do another reduction of NAD plus, so turning it into NADH, into succinyl-CoA, which is 4 carbon attached to coenzyme A, and then ADP plus phosphorus will turn into ATP, and turning it into succinate, which is 4 carbons, and then FAD will turn into FADH2, which turns this into fumarate, the 4 carbon molecule again, and then malate, 4 carbon molecule. We reduce NAD into NADH, and then we get back to oxaloacetate. That's the Krebs cycle. Coenzyme A goes in and out of succinyl-CoA for that ATP plus phosphorus reaction, just so you know. So just to recap the Krebs cycle, this happens twice simultaneously, right? Because we still have 2 pyruvates that are going through this cycle. Oh, what was that? Okay. But we have but we have 3 NAD reduced, 1 ATP produced, 2 CO2 released, and 1 FAD used in each cycle. And then from the Krebs cycle, we go on to the electron transport chain. The final step, if I can get there in my notes. I'm gonna have to cut this, I think. Found it. Okay. So the electron transport chain is confusing to me, but I'm going to try to explain it the best I can. Basically, we have NADH plus H plus unreducing, so oxidizing, by bumping into NADH dehydrogenase. And by that mean dehydrogenase means that it's going to remove the hydrogen or the 2 electrons and then pump a proton through. So it'll pump a proton through into the inner mitochondrial membrane and the intermembrane space. It could be either the H, like the H proton that was removed. It could be either H. It is pumped into the intermembrane space by the enzyme through active transport, which is against the gradient. Then FADH2 has 2 electrons removed, and then it turns to FAD by the Q enzyme. Then the 2 electrons get pumped through by the second proton transport protein. It does the same thing as dehydrogenase. It pumps through the proton through the to the intermembrane space. Then cytochrome C is in the chain, and then 2H plus half an O2 will turn into H2O, and through that the enzyme is still there, so it'll bump in, and then 2 electrons are taken and pumped through as an H plus proton, and it's the same thing again. It's basically a big conveyor belt of proton pumping. It's a series of redox reactions, and then in this case the protein is an enzyme, just so you know, and the water is like a final electron acceptor, so it's used to reduce O2, which creates H2O. And then down the electrochemical gradient will the protons go, and then they'll go to ATP synthase. ATP synthase allows protons to flow through chemiosmosis. So as it's flowing through chemiosmosis, the force of repulsion between the H plus and the ATP synthase turns the enzyme and creates active energy for creation of ATP. And the force of repulsion between H plus and HTP is actually called proton motive force. And the enzyme is separate from the electron transport chain, so it can be anywhere except in between proteins. And yeah, so from there, we've produced, in our total numbers, or 8 NADH plus H plus into 8 NAD, 4 FADH2 into 4 FAD, 24 H2 H plus plus 6 O2 equals 12 H2O and 24 electrons. So 16 electrons, 8 electrons, and 24 electrons were used up. And then if you add that together, it's not 32, but we end up with 32 ATP somehow. That's just like how many protons are used up and pumped through. Yeah. Yeah, because 8 plus 4 plus 24 is 32. And that is done through oxidative phosphorylation. We will have ADP plus 32P turn into ATP through the ATP synthase energy. So all in all, we end up with 36 ATP. And that is that. Then we move on to photosynthesis. So the equation for photosynthesis is 6 CO2 plus 6 H2O equals C6H12O6 plus 6 O2, which is 6 carbon dioxide plus 6 water equals glucose and 6 oxygen. And for that to happen, you must have light, chlorophyll, and enzymes. Not all autotrophs will do photosynthesis, however, most will. So just to go over the brief biology of a plant, we have the cuticle, which is the waxy, shiny surface that prevents water loss, the epidermis, which is the outer skin, which is clear so sun can go through, the mesophyll with, oh, and it has like not many organelles. The spongy mesophyll, no, sorry, the palisade mesophyll, which is like tightly packed together and has the most amount of chloroplasts, and the spongy mesophyll, which is a little less chloroplasts, but both do photosynthesis, and the lower epidermis and the vascular bundle inside the spongy mesophyll, and then the stomata with guard cells at the bottom. And then inside you have a vacuole and a cell wall and chloroplasts. And inside the chloroplasts you have an inner membrane, an outer membrane, and a stroma and thylakoid discs. So photosystem 2 is the first stop for the photons to go through. So there's a variety of pigment molecules with different chlorophylls, and lights are absorbed there. And then it captures the light energy, and then the energy excites electrons to a higher energy level. So these high energy electrons are passed through the electron transport chain, or the ETC for short. To replace the lost electrons, water, H2O, is split in photolysis. So that's 2H2O becomes 4H plus 4 electrons and O2. This produces oxygen gas as a byproduct. Then, through the electron transport chain between photosystem 2 and photosystem 1, the electrons move through the protein complexes in the thylakoid membrane, and as the electrons move, protons are pumped from the stroma into the thylakoid lumen. This creates a proton gradient, high H plus concentration inside the thylakoid. And then in photosystem 1, electrons now at a lower energy arrive, and then the light energy re-energizes the electrons again. High energy electrons are passed to NAD plus, P plus, reducing it to NADPH. So NADP plus 2 electrons plus hydrogen equals NADPH. And then there's ATP synthesis through chemiosmosis, which we already learned about in cellular respiration. It's the proton gradient created earlier drives ATP synthase. The H plus ions flow through the thylakoid lumen back to the stroma through ATP synthase. This powers the formation of ATP from ADP and phosphorus. So ATP is kind of like our energy currency. It's like what's produced. And then NADPH is the electron carrier with reducing power. And then oxygen is the byproduct from splitting water. And that's the light dependent reactions. Now there's the light independent reactions, which are the Calvin cycle. So in reality, it's 1 CO2 at a time and 3 CO2 minimum for product. But for our purposes, there's 6 CO2. Yeah, like for our purposes, it's like 6 CO2 going in. But just know that the product in reality is half a glucose. It's not a full glucose. Um, so phase one, carbon fixation. You have 6 RUBP, which is 2 phosphorus and then a bunch of 5 carbons. And then 1 CO2 is dropped in. And then this turns us into eventually 12 3 carbon molecules of the phosphorus, which is known as 3Pg. So like they're all dropped in eventually and then they turn into 12. But at the start, it's just like one at a time. It trickles in. And then we move on to reduction, which is we'll have 12 ATP turn into 12 ADP, providing another 12 phosphorus for each molecule, 1 phosphorus for each molecule, or 12 phosphorus in total, which is 12 phosphorus, then 3, it's 1 phosphorus, then 3 carbons, and 1 phosphorus at the end, 12 times, which is known as BPG. Then 12 NADPH plus H plus are oxidized into 12 NADP plus and 12 phosphate. And then that turns us into 12 G3P, which is a 3 carbon molecule with a phosphorus, which is a sugar now. It's not an acid. And then as an endergonic reaction, some, like 2 G3P, will turn into 1 glucose and other organic molecules for the phosphorus, but we'll also have 10 G3P keep going through the cycle. And now the 10 G3P is like a 3 carbon molecule with a phosphate at the end. And then 4 phosphate will come off. 6 ATP will come in, turn into ADP, giving us 6 phosphate. So then we have 12 phosphates left, and it goes back to 6 RUBP, and then that's the Calvin cycle. It'll just keep going. So for photosynthesis and the environment, like there are some limiting factors. So temperature, light intensity, and carbon dioxide concentration are limiting factors. So as leaf temperatures increase, photosynthesis goes an exponential rate, increases at an exponential rate because enzymes work more efficiently. But at a certain temperature, photorespiration begins. We'll get over what photorespiration is in a second. But at some high temperatures, denaturation will stop the enzymes. So then it'll go down in efficiency. Then for light intensity, the higher the ATP plus NADPH plus H plus is influenced by the higher the light intensity at the start. But then it becomes limited by the CO2. And then you damage chlorophyll 2 molecules at high light intensities because of photooxidation. So then it's not as efficient and it still decreases in efficiency. And then for carbon dioxide concentration, collisions between the enzyme and the substrate make it an interesting one. At the start, carbon dioxide limits rate of light independent reactions, and lower levels in the atmosphere will like limit this. But then enzymes will be saturated and then it'll just plateau. It's not going to decrease the efficiency at any point, but it will plateau. So what is photorespiration? Photorespiration is like when it's hot and dry outside, the stomata close to conserve water, but as a result, CO2 cannot enter the leaves. So instead of using CO2 for photosynthesis, you use oxygen for photorespiration. So oxygen will go and turn into rubisco, but then you end up with 3Pg and 2 carbon waste, and ATP and NADPH and 3Pg and ATP and NADPH and RUBP. So the cycle will still happen, but it is a lot more costly. Like it uses a lot more NADPH. So not great. 2 ways to combat photorespiration: limit water loss and increase CO2 concentration as an acid inside the stroma. So that's what plants do. And there's always C4 photosynthesis, which is when it's like done between 2 cells. So PEP will turn to oxaloacetate, which you remember from the Krebs cycle. That's like the 4 carbon ingredient at the start. And it'll go turn into another 4 carbon molecule. And then that'll go into the bundle sheath cell where the Calvin cycle will happen. So basically the light dependent reactions, CO2 would leak out of the leaf if stored as a gas, so CO2 is stored as an acid. And then this happens in the mesophyll cell, not palisade or spongy, it's just mesophyll in C4 plants. Um, and initial carbon fixation, so storing CO2 with PEP and carboxylase. That's what happens. So this is just like light independent. Sorry, I mixed it up. So one ATP is used and turned into AMP. This is higher CO2 plus Rubisco, lower O2, just so you know. And then there's CAM plants. CAM plants, these are the ones that are like doing photosynthesis at day and night, different steps. So at night, they store PEP as oxaloacetate and C4 as like a carbon 4 molecule and CO2. And then in the day, CO2 will go into the Calvin cycle and then produce G3P. And yeah, that is it. So what is cell-to-cell communication? It's basically just how cells talk to each other. So signaling, response, and regulation. So what is homeostasis? Homostasis. Homeostasis. Jesus. It's constant internal conditions. So is the maintenance of homeostasis within a cell, body, as the environment changes. The variables are all intertwined with each other. So blood, pH, and CO2, and breathing and O2, like these are all intertwined and they all influence each other, and homeostasis cannot be achieved if you do not have constant internal temperatures for all. So it's actually a really hard job that your body does. So some examples of some constant internal conditions that need to be maintained: carbonic acid, temperature, pH, ATP production, vitamins, cholesterol, water, osmolarity, tonicity, blood pressure. Osmolarity is the balance of water between two cells within a membrane, and tonicity is from perspective of solute. So there are four mechanisms of communication between cells. There's contact communication, which is like local one-to-one conversation, basically like a one-to-one conversation between cells. So really done by proteins, like the messages are proteins. And then it's like calcium in muscles and immune cells. And then there's paracrine signaling, which is like a presentation to those around you. So something is secreted, exocytosis, and signals sometimes to itself, which is autocrine. And then there's synaptic signaling, which is like a phone call between two separate cells. So the gap is the synapse. And this is usually done through neurons. And there's endocrine signaling, which is like social media, or like what I'm doing right now, spreading misinformation. It's releasing the crine or something in blood. It's exocrine if outside the body. And then there is negative feedback loops, which is what influences homeostasis. So there's a response. And then, no, sorry, there's a stimulus, which is like the reason, the problem. And then it goes to the receptor, which is the stimulus acceptor. And then it communicates with the processor, which communicates with the effector, which is the signal. And then it causes a response. And it usually does something in opposite to of stimulation. It's just the opposing trait in response then to the stimulus. So this is usually done through G protein coupled receptors. So there's molecular specificity between a hormone and a receptor. So basically it's like lock and key. So there's only one hormone that matches to this one receptor, but the hormone will bind to the receptor protein on the cell membrane. The hormone is also known as the ligand. And then the receptor changes state from GDP, the G protein, when inactive it's GDP, will get kicked out and replaced with GTP in step 2. So it turns into a GTP, and then that G protein becomes activated G protein, phosphorylates exenyl cyclase enzyme to activate. So yeah, the activated G protein will phosphorylate acetyl cyclase, adenylyl cyclase enzyme to activate. So ATP will turn into CAMP, which is the secondary messenger. And then that'll cause a cascade reaction from inactive messengers to active, inactive to active, inactive to active. And the advantages of this signal transduction is like a small signal is amplified in response if the molecular pathway activates subsequent molecules. The small size of the secondary messenger means it can diffuse in the cytoplasm, so large molecules can't really do this, but this can. And then multiple enzymes will turn on, and you can go back and stop if one step if the error occurs, because there's multiple steps. Then we move on to the endocrine system. So hormones are chemical messages, some proteins, produced by endocrine glands. They travel through the blood and maintain homeostasis through negative feedback loops. Two examples of these hormones are insulin to glucagon, and then there's also the thyroid, parathyroid. Sorry, the pancreas has the insulin and the glucagon, and the thyroid has parathyroid. And then the pituitary gland, we will get to what it has. There are protein hormones which bind to receptors in the cell membrane through signal transduction pathways, and they cannot go through the membrane. And there's lipid hormones which easily cross through membranes, such as the phospholipid bilayer and cytoplasm receptors. So lipoproteins, they're also lipoproteins because they're transport proteins, but protein hormones bind to receptors embedded in the cell membrane. But nothing happens even if receptors are in the membrane or the cytoplasm, sometimes if they reach non-target cells. Then there's the plasma membrane. Then there's the lipid hormones where the plasma membrane, they will pass through the plasma membrane. And then inside the target cell, the hormone binds to a receptor protein in the cytoplasm or nucleus. Hormones receptors complex bind to hormone response elements on DNA, regulating gene transcript. Then protein is synthesized. Then protein synthesization increases or decreases is the cell response. There are endocrine organs, but the main ones that we'll focus on today are the hypothalamus, the thyroid gland, and the pituitary gland. So in the hypothalamus, that's like the hormone producer. Sorry, I just saw an image and I got confused. But like an example of negative feedback is, yeah, we'll go with this one. So the stimulus would be low blood temperature. And that goes to the receptor, which detects by thermostatic cells and sensors in the skin cells. That goes to the hypothalamus as a signal. Then TRH hormone will go down and make the anterior pituitary tell, like the TRH will tell it to produce TSH, which will go to the thyroid, which is told to produce T4 and T3. And then T4 will also go to the hypothalamus, and then it's a whole convoluted loop. I'm not a doctor, but then it'll go to the effector, which is the muscles, and then that will go to cause a response of an increased metabolic rate, which creates heat, and then we are no longer cold. Another example is dropping blood glucose will trigger glucagon, which then triggers glycogenolysis and fat breakdown, which is basically what that is, which then causes rising blood glucose. But if it rises too high, then insulin will be triggered, and that will trigger glucose uptake, glycogenesis, and fat storage. And then that'll cause dropping blood glucose. But dropping blood glucose can also be caused by exercise or starvation, and rising blood glucose can also be caused by food. Negative feedback with a time delay sets up oscillations about a set point, so like it'll wave. So when you eat and you get insulin, it'll raise, but then it'll go down, and then to raise that, it'll produce glucagon, and then it'll go back up, and then you'll produce insulin, and then it'll go down, and then it'll produce glucagon, and then it'll go up, and then insulin will go down. But the first wave is always really big. So when you eat something high in simple sugars, then you'll crash. Yeah. Then we move on to the kidney, which, living things produce nitrogenous wastes. The three nitrogenous wastes that an organism produces is ammonia, amino acid, ammonia, or uric acid. Sorry. Oh my god. Ammonia, urea, or uric acid. So fish will produce ammonia, mammals will produce urea, and birds and reptiles will produce uric acid. And this is an order of like toxicity. This is an endergonic reaction. So I'll throw up a picture of a kidney on screen. But there's like a cortex, which is the outer layer and filtration of bodily fluids. There's the medulla, which has charge of water recovery. There's the pelvis, which collects urine, and there's the renal artery and vein. Now, inside this medulla, there are these things called nephrons. What is a nephron, you might ask? I'm glad you asked. It is what filters urine. It's what filters the water, the blood, and all that stuff for wastes. So we start with the glomerulus, which is like the blood comes in, the blood filters through gaps in the capillaries, and small solutes in water get through, and then proteins and cells stay in blood. Then it filters into Bowman's capsule, which collects the fluid excreted from the capillaries from the glomerulus. This filtrate goes through the paroxysmal tube, but it shoots glucose and amino acids back into the blood. Glucose, amino acids, sodium all are active transport, and then chloride and ions and H2O are passive transport, and those get filtered through. And then it goes to the loop of Henle, which is the ascending limb. The descending limb, H2O will move, and then the ascending limb, only sodium will move. And then it'll go to the distal tube where sodium goes into the blood, and same with H2O through passive transport. Then potassium, hydrogen ions, and HCO3, creatine, drugs, etc., go out of the blood through active transport and into the urine. So there's like low salt. So then we go to the collecting duct, where the top has low salt, and then the bottom has like a higher concentration of salt. So water must be recovered into blood. Water facilitated diffusion channels, so aquaporins increase and are produced. So the concentration of urine is controlled by hormone ADH from the hypothalamus into diuretic hormone. ADH is the antidiuretic hormone. Technically, this happens. Technically, the posterior pituitary, it's whatever. ADH ups the amount of aquaporins open, and then alcohol and caffeine are diuretics, just as a side note. So when you take them, it makes you pee more because it's higher in water concentration. You need a high volume of water in the body to survive, and you need a low volume of blood in the body to survive, but you still need a volume of blood. So you have so much volume of filtrate produced daily, but then it's recovered by osmosis, and the volume of urine excreted daily is like this much versus this much. So just so you know. The nervous system has two major cellular components: glial cells to help support neurons in a variety of ways, such as physical support, supply of nutrients, produce myelin, and neurons are cells that can transmit an action potential or an electrical impulse. These cells make up two branches of the vertebrate nervous system: the CNS, central nervous system, consisting of the brain and the spinal cord, and the PNS, the peripheral nervous system, consisting of all the sensory and motor neurons. So sensory neurons registering internal stimuli, that's like a full stomach. And then that'll go into sensory pathways, which will go to the brain and spinal cord, which will go to motor pathways, which go to the somatic nervous system or the autonomic nervous system, which is involuntary. And then from the autonomic nervous system, then there'll be like parasympathetic nervous system, rest and response, or rest and digest, and the sympathetic nervous system, which is fight or flight. So muscles in the arteries, food through the digestive tract, all that stuff, that's the autonomic nervous system. A neuron is a cell that has the potential to pick up chemical or physical signals in one location, to integrate those signals, the chemical decision on if there's enough signal to make a signal, to carry a signal electrochemically along the cell to a target location, through neurotransmitters to secrete a new chemical signal that can diffuse to a target cell. And each of these functions is a specialty and different parts of the neuron. I'll throw up my picture of the neuron right here. We're not going to go over the anatomy, but just know there's like a myelin sheath, there's axon terminals, there's an axon, there's a nucleus, there's a cell body, there's dendrites, all those lovely things. Ultimately, the function of neurons depends on the electrical activity that is driven by the movement of ions across the cell membrane. So it's like more negative inside, more positive outside, and transport proteins make sure of this. So there's types of ion transport proteins in our membrane, such as passive channels, so like sodium and potassium, which is facilitated diffusion. There's gated channels, which make diffusion go from high to low. So voltage gated channels, doors open and close based on the voltage for sodium, potassium, and calcium. Chemically gated channels, doors open and close based on the chemicals present. And then mechanically gated channels, which we don't need to know. There's also ion pumps for active transport, which is sodium and potassium pump. Yeah. So I'm not going to go over this in too much detail. You can go over it on your own time. But I'm just letting you know that there is less potassium outside the cell and less sodium inside the cell. So more potassium inside, more sodium outside. Okay, we'll go over repolarization. So number 1, action potential is -70. Resting gates are closed, voltage gated channels. Number 2, local cytoplasm reaches -55. Stop with the balloons. -55 milliliters. Sodium gates open. Sodium rushes in. Then sodium pushes neighbor cytoplasm to -55 millivolts. I've been saying milliliters. I mean millivolts. Millivolts threshold, so more sodium gates open. That is depolarization. And then sodium gates close at its peak and potassium rushes out of the cell. That is repolarization. Then hyperpolarized membrane by the slow close of potassium and slow close of potassium gates, and sodium gates close. Neuron stays in refractory period. So like the neuron can't send action potentials at this point. And then it gets back to like the start of where it's supposed to be, and then the cycle begins again. And that's the unit. You did it. You finished. Grade 12 bio. I'm proud of you. You did amazing. And no matter what grade you have, like, okay, I don't know if I'm crazy or if these things keep popping up, but I don't know what photo booth's doing. But point is, congratulations on being done grade 12 bio. Sincerely, I wish you the best of luck on your exam, and you'll do great. Thanks.